Hi, Sean. Thanks for joining me today. I really appreciate it.
Hi, David. Pleasure to be here. Thanks for having me.
To start, would you be able to introduce yourself to the audience and talk about your role at Phenomenex?
Sure. So my name is Sean Orlewitz. I started with Phenomenex a little over 22 years ago. Yes, I was 12 years old at the time. I've had the very pleasure and fortune to have many roles at Phenomenex, both in our labs, outside of our labs, in our clients' labs. But today, and for the last year or so, I've been the principal marketing development manager for our pharmaceutical sector. What that really means is I'm one of the leaders of our strategies around what we do for our pharmaceutical customers.
And a big focus in that area that we're going to get into today is the GLP-1 work.
What are those? I've never heard of them.
What are the
I'm kidding. Obviously, yes, this is a very hot area in pharma, and it has been for quite some time. But I think the commercialization and popularization of a few drugs that are out there in the market today have made it really a household word.
So, in terms of your role, was that just kind of a right place, right time situation in terms of how you became involved with it?
I hope not. I'd like to believe that I have some role in it. And so, as I mentioned, I've been around Phenomenex and really the areas of chromatography and the related sciences for quite some time. The way that my career has maybe progressed was somewhat serendipitous in that previous to this role, I was focused on the North American market for generic pharmaceuticals. And so we've really put a strategic effort behind helping the generic pharma customers bring drugs to market faster, better, more efficacious, things of that nature, really because we see a benefit in more affordable medications worldwide. And so that was something I was really passionate about for quite some time. I don't know, maybe 10, 13 years ago, I had come across my first GLP-1 and exenatide when I was in the lab. But really where I started to cut my teeth and get my real introduction into GLP-1s was the genericization of liraglutide. So I started seeing this, I forget the exact dates, but I want to say Lira or Victoza went generic in 2022 or 23. So the years preceding that, it was what everyone was working on. It was what everyone was doing. And it was one of the first peptides that I saw going generic. So we were working with both the synthetic peptide manufacturers, their DMF filings and their methods there, as well as all of the drug product manufacturers and their ANDA filings. And so everyone was really developing methods around peptides and their related substances for that. And that's really how I got my start. Now moving forward, as I now have a global role, we're seeing the maturity of the market, both from an innovator side with, drugs like Tirzepatide and Semaglutide, and then the next phases of genericization with Semaglutide, which is starting to go generic in some regions as early as next year. So this popularity and then movement from innovator into generics, and then all of the aspects around that, Dave. So, Phenomenex supports clients all the way from API manufacturing. And fortunately for us, most of these synthetic peptides are purified by reverse phase chromatography on a commercial scale, all the way down to the analytical workflows that test purity and aggregation and things of that nature. So I feel like we have a nice view from multiple lenses of the GLP-1 industry.
And that's a great segue because the next thing I wanted to ask you about was what are some of the main analytical challenges with the separating and the quantifying of those analogs like semaglutide?
Yeah, so peptides, are this, again, some people want to compartmentalize pharmaceuticals into large molecule and small molecule or into pharma and biopharma. And I think there's a lot of verbiage that's out there. And I don't know as a chemist if I really care what you want to call them. I think of Romeo and Juliet and, a rose by any of the name. But peptides really do fall in the gray spaces in between all of those terms, right? Are they small molecule? Well, they're very manageable from a small molecule chemist like me in terms of the number of residues that we're looking at. They're really just a combination of amino acids, but they're also very specific and targeting, kind of like biopharma is going after from a therapeutic area. So I feel that they have that gray area. But really, again, as the chromatographer, the challenges are really unique in the fact of the related substances, whether they be manufacturing, process impurities or degradation products, are so closely related, like structurally, chemically, all the way down to a molecular formula is the way that I look at it. So peptides are these large conglomerations of amino acids, a lot of their impurities are a deletion of one amino acid or one oxidation at one position, or God forbid it's isomerization, where it's the same exact molecular formula, just in a different shape or something to that nature. And so they're so chemically related that it's a little bit different than what we're used to when we're talking about like a substituted nitrobenzene, which is a pretty different structure. And so we can use the different modes of chromatography to get good chromatographic separation, where if you think of a large peptide, let's just make up a number like 31 amino acid residues, and it's got one less amino acid, relatively that's a very small change. So I think it's those really small changes and small differences between the related substances which make them very challenging to separate.
For our audience of chromatographers, what would you like people to know about the Ares peptide XBC18 column?
So yeah, I'm guessing you bring up Ares XBC18 because it's been widely referenced in literature. It's been our go-to peptide column for reverse phase for quite some time. And even expanding it, I can get into the why. Why is that popular? It really, there's many factors in chromatographic separation. There's many variables, but I will narrow this conversation for peptide residues and for related substances and degradation products under reverse phase conditions. There's really two variables that matter the most in terms of chromatography. That is efficiency and selectivity. I'll talk about the second one first because I think it's the harder to wrap your head around as maybe a chromatographer. I am a chromatography geek. I have been for more than 20 years. And more so, I work for a company that only manufactures columns. Now, I love instruments. I want to be clear about this. In my hobbies, I work on motorcycles and cars, and I love working on instruments. But my career is focused on the surface chemistry of chromatography columns. And that really affects selectivity or alpha in the resolution equation. And that selectivity it's hard to quantify, but it's really there. And so going back into the separation, typically speaking, reverse phase separations are based on hydrophobic differences. We use C18s to separate molecules that difference by their carbons. That's the fundamentals of reverse phase chromatography. Van der Waals forces, carbon-carbon interactions. Now, backing what we just talked about, Dave, the differences between these peptides and the related substances are amino acids or oxidations. These are more ionic or polar in nature. And so it's these secondary or non-van der Waals forces type separations that make the difference in the ability to separate these compounds on a chromatographic column. So that's what I refer to as selectivity. And this is why every manufacturer has dozens of C18s in their catalog. They're not all equal. They all have slight differences in selectivity, and it's those secondary mechanisms that we control. Now, why Ares? Years ago, we did some work on peptide mapping. So a little bit of a different application, but we were taking protein digests and taking a look at peptide mapping, and we knew that Ares XB chemistry had a very unique selectivity for peptides. Fast forward years later, we were very happy to learn empirically that same selectivity advantage benefited synthetic manufacturers as well. So that was kind of the selectivity story. Now the extra, I don't know what you want to call it, cherry on top is my second variable in the chromatographic equation, which is efficiency. Empirically, in my experience, efficiency plays a huge role in these peptide separations because, again, they're very small differences. So when I have very small resolution between two peaks, and also let's use a drug substance as an example, the main API peaks there at 90-something%. My small related substances there at 0.5%. If my peaks aren't very symmetrical and very tall, they're not very efficient, I'm not going to be able to accurately integrate them. I'm going to manually draw baselines. That is going to lead to precision problems. We knew that efficiency matters in these separations because adding efficiency, it gets that extra boost in resolution that allows you to integrate really well and have a robust quantitative method. Ares is built on a core shell particle. These are these superficially porous particles that many of us have that are really just aimed at efficiency. That's their whole purpose of the core silica particle, the foundation that the Ares XBC18 chemistry is put on, the way I look at it. We have the selectivity in the XB chemistry, and then we put it on a really efficient particle that not only is efficient, but also scales from traditional HPLC, 400 to 600 bar systems, to we can go sub 2 micron and go to, UHPLC type applications to really maximize that efficiency. So, there's a lot of factors in that, and we can chromatographically geek out for hours at a time. But I look at it and really, those are the two key factors in terms of why those chemistries do the best for these types of separations.
Reproducibility is always important at some level, right? But I have to think that for GLP-1s, therapeutics, the regulatory environment and what's required has to be at the highest level.
Yeah, these are under the microscope, right? These are all of us are hearing about these daily. The usage, the population that are benefiting from the use of these therapeutics is massive. If you look at the future, if you look at what some of these companies are speculating in terms of what's going to happen between now and 2030, yes, again, so the safety and efficacy of these drugs it's paramount, right? It affects a huge population. Then throw on top of this that for the majority of these products, they're sterile injectables. These are widespread injectables. This is not just another pill that benefits from the GI tract. Now, that might be the future. We'll talk about that later. But for right now, these are injectable products. And so this, sterility, transferability, shipping, user error, again, putting sharp things in front of large groups of human beings, not always a successful strategy. Anyways, so yes, they are under the microscope. So now, what does that mean analytically? For me, it means a lot of things. Number one, to your point, the methods must be robust because, as we talked about, the impurities that may affect the therapeutic benefits, that may affect the bioavailability, that may affect the toxicity of these drug products are very small. And the differences, like we just talked about, are little. So the chromatographic resolution and separation is really important and needs to be robust. Second of which, because of the scale of it, you got to imagine these methods are being transferred all over the world. I've seen this personally. Phenomenex, I'm very fortunate, as I mentioned, I have a global position, and a lot of our partners are global companies. And so I'll get one phone call from Indianapolis saying, hey, can you jump on a conference call with our partners in China who are trying to run that same method that we run here? And that's the reality of the game. And we all want to pretend that methods are completely transferable, but for any of us that have actually worked in the real world of method transfer, they're not. The analogy I like to use, Dave, is recipes. So we make our SOPs and our methods and everything we do are recipes. And we do our best to write down the ingredients and how much, and we really do a good job. But if you give a great recipe to a bad cook, food's not going to taste very good. And so that's really method transfer. There's still like a skill level on both sides of this that we have to acknowledge. And so yes, in building robustness up front, in your method development, in your validation protocols, you ensure that there's at least some mitigated risks in that method transfer. And again, you better get the same results with all of your channel partners and CDMOs and CROs all over the world. And so yes, and then furthermore, for these types of products, they're complex, right? Just to use some examples, I would say in the typical small molecule oral tablet USP monograph, you've got somewhere between 2 and 6 impurities, pretty commonly called out. I just, from a lifetime of running monograph methods. In the latest method that we did with semaglutide, I believe we have 13 known impurities. And that's just scratching the surface. So the samples get more complex and they get harder. And so again, you really need to build in that robustness up front. And then furthermore, these methods they're tough. They're often very long. I hate to say it, but I like short methods. And there was a time in my life where I was trying to get like the 1-minute method. These are not that. These are complex scenarios. I often see 45-minute methods. So that means shallow gradients. And anytime I see a shallow gradient, I see an instrument to instrument issue. Because now you talk about instrumentation challenges where if one instrument has a different gradient delay volume than instrument B, when you have a really shallow gradient, those retention times will be different. Different flow cell volumes. Instrument manufacturers are great and they're complex and the instruments are better than they've ever been today. But with more options becomes, differences. Somebody might have flow cell A and somebody might have flow cell C, and they're wondering why their peak asymmetries are different or their peak volumes or their peak heights are different. So To your point, with more complexity comes more challenges and more considerations that you have to have around robustness. But I really do, especially for the audience here, I encourage you to spend the time up front during development and during your validation to ensure there is robustness. Talk to vendors like us. Get 3 batches of columns. Test them all. Make sure you're using good mobile phase reagents. Make sure that you're considering the instrument aspects of it around gradient delay volumes, around system dwells, about injectors, flow loop injectors versus flow through. I mean, all of these parameters really add to robustness in these complex methods. But overall, it's not easy. But we've done it. I think a lot of people have been successful. And again, the other thing, Dave, that this brings in is GLPs are so popular. These are some people's first peptide. These are small molecule people, quote unquote, that are getting into this world. And so I think that getting an introduction into the increased complexity is really important.
That makes a lot of sense to me. On the instrumentation side, my world is a lot of LCMS, MS.
Yeah.
My company's doing the sample concentration for those samples post-extraction. So I don't necessarily consider it an emerging tool, but certainly continues to grow in popularity.
Yes.
I was curious how a high-res mass spec could transform the analysis that's being done in the GLP-1 space.
I've kind of had different roles in my career, but one of my roles was very focused on mass spectrometry for a couple of years. And so I always kind of joke around, let me tell my joke first, that there's kind of 2 people in a mass spectrometry lab. There's the chromatography that only looks at the mass spec and sees a detector. And then there's the mass spectrometrist that looks at the HPLC and only sees an injector. So try to be both. And the role of mass spectrometry in GLP-1s is significant. And really, again, it's two-sided. The easy thing to focus on is the role of high-resolution mass spectrometry in research. These therapeutics were developed and took off to address diabetes, is where it started. And then it naturally progressed into weight loss because of the results of the clinical trials, both done in clinics as well as in the real world. Well, we're seeing huge opportunities in other therapeutic areas now. And I think there's a little bit of backwards movement of using high resolution mass spectrometry to understand how these specific drugs are targeting proteins and receptors that are beneficial to other therapeutic areas. And so we've seen a lot of that being done. And it's really kind of fun because, I don't know, maybe cut my teeth in early discovery and high res going the other way, looking at the receptor or looking at the target and trying to design a molecule that fits it or something to that nature, where this is the other way around, right? Here we have something, and so what else can it do? And it gets into like really the benefits of therapeutic peptides and how specific they can be, which aids in maybe not having as many side effects and those other things. And so that work is being done at lightning speed, as you're seeing a lot of NDAs and R&DAs going out there for other therapeutic areas like addiction and many others that we're seeing. Now, the more interesting thing that I've seen, and again, this goes maybe sharing a little bit about my personal growth, is in the global market. So Dave, you and I appear to work in North America, where mass spectrometry has been prevalent and been growing for quite some time. But when you go to the emerging markets like India, it's an expensive technique, and it's not maybe as popular as it is today. But these molecules, and especially from a process analytical standpoint, all these challenges that I mentioned about shallow gradients and method transfer and small differences in the molecular structure can be solved by using mass spectrometry. As a chromatographer, as I just really went off on, it's very challenging for me to separate chromatographically a 31 residue peptide by the position of a lysine. Like, that is hard work. You know what a triple quad mass spec does really well? It separates them in the spectra. So that's where clients are starting to adopt that technology because of its specificity, right? And that's where I really see the benefits and the growth from it. And so I think there's a process analytical benefit to mass spectrometry. I also think that there's a quality control aspect of mass spectrometry because, again, if you're looking for an oxidated product, and again, back to don't tell Phenomenex, but do you need to chromatographically separate that? It depends, on the ion suppression situation, but chances are you might be able to use the triple quad in order to get that separation and do it very robustly at a very, very low limited quantitation. So I think that there's a huge role in it. I look forward to seeing its prevalence and its utility moving forward.
It's been so interesting to me to read over some of the materials available on the Phenomenex website because it's just so different from what I deal with on a daily basis, where it's more EPA methodologies or at the academic research level work that's derived from those methods.
Yeah.
Where it couldn't be more different, I feel like, in terms of how strict things were, especially the people executing at the EPA method level. It's nope, this is just what we do, and we just purchase the materials listed on the standardized method, and off we went.
Yeah, I mean, that's always been a unique aspect of my career, is sort of gauging the boundaries by which each client is operating within, right? And so it's, it's a breath of fresh air when you talk to somebody that's in like pure discovery, where they're like, I could use anything, and I have every detector, versus, and again, to use this analogy, go to a quality control environment in a generic pharmaceutical company. This is the flexibility that they have. But at the same time, we have to work within those boundaries, and we have to be empathetic to them as well. So they're there for a reason. And usually the reason is safety and efficacy. And I support that 100%.
Yeah, it's critically important. There's no getting around the safety aspect. You've touched on this a little bit, but are there any common pitfalls or troubleshooting tips you would share with an analyst who's working on these peptides?
Yeah, there's lots. I mean, overall, we'll stick with analytical workflows. I think, and again, I'm going to put this through the perspective of we support clients globally in all situations, and luckily we've been around for a long time, so we have a lot of good relationships. And I can share that GLP-1s has been the path of growth for many companies, both in innovative pharma and in generic pharma, in terms of their entry into peptides, their entry into sterile injectable products, their entry into size exclusion workflows. So for peptides in a sterile injectable formulation, the FDA requires the drug product be tested for aggregation. And this is commonly a size exclusion workflow, in which you're looking for the formation of the monomer peptide to a dimer and trimer in solution under shipping conditions. Is it stable on a shelf at the end of the day? Because as it dimerizes or aggregates, its potency becomes significantly decreased. And in some cases, that dimer or trimer could actually become therapeutically harmful. So this is size exclusion chromatography, not reverse phase, not gas chromatography. So we've gotten phone calls recently in the last couple of years. People have never ran size exclusion chromatography, so they have a lot of questions. And so I think that, and that's the same thing with maybe reverse phase residues. My first advice is just Google. Use the Google. It's a wonderful tool. Lean on vendors like Phenomenex and the others that are out there that have done this before. I managed laboratories in my career, and one of the biggest things I learned is learning what not to do from others, learning from other people's mistakes. There may not be the exact solution for your unique problem out there in the Google, but there might be some things that I wanted to try that, but don't try it, it doesn't work. And I think those are the conversations that are really useful. Be patient. These are, again, and this is just more of me projecting about the conversations I've had over the last 5 or 6 years with people. Again, if you're small molecule type drug product manufacturer and you're getting into these drugs, you're used to 10 to 20 minute methods and walking away. You're going to have to be a little more patient with these types of methods, and that they're typically 45 minutes, and in some cases they're 100 minutes. And it sounds like it's going backwards, but it's not. We're using very high efficient particles, often on UHPLC type instruments at high pressures to get massive efficiencies. And we're using modern technologies. And so that would be my other thing, is that, talk to everyone, talk to anybody you can, go to conferences, look at, the search. What's really kind of alarming and interesting to me, specifically to GLP-1s, is there's not a lot of peer-reviewed literature out there. I think that these were a little bit homegrown and innovator type companies where they didn't want to let all their secrets out. And so even, myself, who tries to keep his finger on the pulse, you're not going to see a whole lot of peer reviewed journals with great methods naming the commercial impurities. Doesn't exist yet. There's not a lot of monographs or compendial methods out there. So you've really got to kind of go out there. And the good news is, for better or for worse, us vendors are very focused on this market because it's a huge growth area for us, and it has been. So, and the last piece of advice I would use is that temperature control of your HPLC method matters. And so again, a lot of people sort of overlook the temperature control of their method as a robustness issue. And that may be true for some methods, but I can tell you from lots of experience that controlling the temperature of your mobile phase and your column for these types of methods is a great way to add some robustness to it. Just a couple tips and tricks, but man, in about 3 years, I'll write a book about it.
Hey, absolutely. That's a great idea. And I certainly don't want you to give away any state secrets in terms of what Phenomenex is working on in terms of product development. But is there anything you wanted to talk about regarding how the company's supporting advancements in this space?
Yeah. Again, I have a loud mouth, and a lot of people out there know me, but I'm also somewhat here to have a loud mouth. This is not new to us. We've known about this market for quite some time. I remember publishing a paper with my name on it in 2013 about exenatide. We have a lot of experience, and so we're applying that experience. I think there's a number of approaches we're taking. Number one, and probably most importantly, is that educational approach. This past year, and specifically this past 6 months, we have published a lot of new technical content on this subject matter. We have worked incredibly hard in our application laboratory to work with real samples too. I'm just as guilty as anybody of publishing a chromatogram with one standard peak in the middle of a gradient. That is not what we're putting out there. We put out a paper recently where we obtained, again, I think 13 commercial and identified impurities that we have found through our insights from our customers are chromatographically important, and talked about the separation of those. And so we're going to continue to do that, and we're going to continue to build workflows because unfortunately, there's no one ring that rules them all in this world, right? I would love to tell you that Ares XBC18 works for every peptide, but I can tell you from experience, it doesn't. We recently published a paper on Tirzepatide in which we found a different chemistry to be more beneficial. And that was based on empirical experience. So, we're humble enough to say that not everything works, but this is also why we have a portfolio of columns at the end of the day. So I think that there's going to be a lot, just not where we're supporting our clients. Education, again, helping these clients that have maybe never worked with peptides or never done size exclusion to adopt those technologies that we have experience with. Again, if they're coming, just using an example, if they're coming from the small molecule world, they're familiar with Phenomenex as well. And so we can help them along that way with some online resources, webinars, educations, on-site visits, whatever they need in order to get there. And then, maybe to peek behind the curtain, we are developing new products for this market. And it's not just for peptides. I want to be clear. We're developing new products for oligonucleotides. We're developing new products for AAVs. That's our responsibility of a vendor, is to be out in front of the technology and understanding where pharma is going. And so, not to plug it, but one of that is very prevalent. This week, we launched a new size exclusion column that is aimed at GLP-1s. It's a molecular weight range. These are really smaller, much relatively smaller peptides that we've seen out there. They're also historically very hydrophobic. I mean, I could tell you from experience, liraglutide is one of the most hydrophobic peptides I've ever worked on. And that's very relevant to its separation, right? So we're doing that, and we're going to continue to do that for the years to come because, again, is not sharing any news. These drugs are not going anywhere. I think they're going to be a story of pharma for the next 10 years. Now, I'm fascinated to hear how they're going to, progress.
To go off on a tangent just for a moment, what always stuck out to me when GLP-1s really entered my awareness was hearing on Bloomberg the talks of what are the second order impacts or second order effects on all sorts of different businesses. For example, if the average customer on Delta loses 10 pounds, what does that mean for them in terms of fuel prices or in terms of General Mills making cereal that people don't want anymore?
Yes. I've been very fortunate to be around pharma for a long time, and I can put kind of pins in my lifetime when certain drugs came out, and GLP-1s is one of those moments because it really is going to be transformative to a lot of things. Like you said, like the foods that people eat. So now another tangent. You and I lived through the end of the STEMI heart attack, right? So I'm not that young of a man, and I come from a very large Irish family. And I could tell you that my uncles, who I have many of, if they passed away in their late 50s from a large STEMI heart attack, was essentially natural causes, right? These are large men who came from the Midwest of the United States who ate a lot of cheese and, yeah, not the healthiest lifestyles I've ever met. Love them to death, but yeah, that doesn't happen anymore. It doesn't. It doesn't really exist. I think I saw something like less than 30,000 STEMI heart attacks globally a year now. And why? Statins. Statins did that, right? And so, and that's one of those kind of pins that you go, holy cow, this had an impact globally. And I do think that GLP-1s are going to have an impact globally, and like you said, in different ways, right? Into those ways. And what's really interesting to me is the direct acute impact on diabetes. I think that's something that's going to be very palatable in a decade's time, and it's going to be fascinating to watch.
Absolutely. My other note was, as a scientific marketer, I can certainly see why, with a lack of peer-reviewed work and possibly, not to make any assumptions, but a lack of customers willing to really disclose publicly all the work that they're doing in the space. The onus is really on you in the applications lab to put all this together.
I hit the nail on it. And again, back to, I've been in and out of our applications lab for many years, and people, that's another thing. Like, people don't know. Do you know how hard it is to get these samples? It is very hard. And so, yeah, to your point, we worked, just to give an example, with that SEMA work we did. A group of my colleagues and I probably worked for 6 months to source those. And it wasn't about just sourcing anything. We went out and got a lot of customer insight about what's chromatographically important. What are the deletions or the oxidative or deamidated, or what are those side chain products that are going to be unique and challenging chromatographically? Because again, it will just use the generic market. So every API manufacturer that's going to file a DMF for semaglutide has a different process. They have different process impurities, right? And so I cannot create a universal application for every synthetic peptide manufacturer because they are all using different process. But I can identify a couple key known impurities that are likely going to happen that are chromatographically important. Because again, if you can, on a C18 with a general TFA gradient, if you can get resolution of 40, you don't need me. You don't need my help. You're good. You know what I mean? So I think that's where maybe there's some collaboration and effort. And that's also where, we are working with the market, because that target is also moving as the market moves. The manufacturing of these products is advancing. The ability for multiple companies to do solid state synthesis really well on these peptides is improving at like a Moore's law rate. So yeah, we hope that the samples become less complex, but I also empirically see it going the other direction.
We'll have to see, and I'll be happy to have you on again to promote that book you'll be working.
Someday.
What else would you like our audience of separation scientists to know about this topic and Phenomenex? We touched on gradient optimization. That's obviously a big giant topic that we could only touch the surface of today. Any other thoughts you'd like to cover?
Man, so much. Where do we start? All right, I'll stick with analytical for a second, because this is an area that maybe we've been focusing on. And that is, I had an epiphany very recently. And I wrote it down, and it's actually sitting above my head, above my screen right now. And I will share it with the team. And that is that the enemy of innovation is the existence of copy and paste. And this came from some interesting things, because everyone's busy. I understand. And we want to copy and paste everything, just reuse things in order to make our lives a little easier. And I'm as guilty of that as anybody else. But I can share from when embarking on peptide separations, don't copy and paste. And what I mean by that is, again, I spent 12 years of my career almost every day in our clients' laboratories. And I can share that worldwide HPLC, reverse phase method development, is approached relatively the same way, which involves my favorite column and the mobile phase that's sitting on the system today. That's where I start. And I agree. By the way, it's where I start. With these separations, I ask everybody, and myself including, to take pause, do some research, talk to some people, because I can share that I've seen some really interesting things in regards to unique organic modifiers. I think TFA is a great ion pairing agent. I think it magically makes peaks seem more symmetrical, but maybe it's not the best organic modifier for all peptide separations. And then the same thing is true for column selectivity. Don't just bang your head up against the wall because your favorite column isn't working very well. It might not be working very well because that particular peptide is very hydrophobic, or that particular peptide is missing a residue that selectivity can't separate. So that would be a topic that I would encourage people to really focus on when it comes to peptides, because the TFA is a good example. It works fine for a lot of things, but it doesn't always work for these separations, and I think that's a limiting factor. The other side that I think is really interesting, and maybe slightly different, but it's still chromatographically focused, is the preparative purification side. Dave, just sharing, I started my career out in large-scale organic synthesis. I loved it, and I still wonder what would have happened if I would have stayed there. We'll tell that story in a future podcast. I did synthesis when I was in my 20s at such a scale that we did a diazonium reaction that gave out nitrogen. The room got windy. Like that kind of scale. So I don't know, like something about the organic chemist background in me. I loved seeing chemistry or feeling it, or feeling the heat on a 200-gallon vessel. Those were exciting times. Preparative chromatography is sort of like today, my version of that today. It's the thing that I get to do that's sort of like seeing chromatography in front of you. It's not just on a screen as a Gaussian peak, right? You've got a fraction and you've got, a rotovaps going. And so most synthetic peptide therapeutics manufactured today are purified by reverse phase chromatography. And I love that. And I know that the people that do it do not love that because it's expensive and it's time consuming and all those other things. But I do love it, and I do see a lot of work being done in that field because it's obviously economically driven, right? Which is kind of nice. It'll get funding. And then I see a lot of opportunity for advancement. When representing Phenomenex, I take some responsibility for this as well, because with as much popularity and as much money, frankly, let's say it what it is, that's behind these drugs, I think there's going to be a lot of investment made in improving those processes, making them more efficient, making the reverse phase sorbents last longer, making those separations more effective. And so, like, just to leave the team, I kind of think of preparative chromatography like a triangle. And you've got cost here, you've got speed here, and you've got purity here, right? Pick two. That's all you get. Then that's sort of right. You can skew the triangle to maybe not so equilateral, but you've really got to lean into one or the other in terms of what you want to accomplish there. And so I think it's a fascinating area about GLP-1s. I think it's also an area that, again, is emerging because API manufacturers, they avoid reverse phase chromatography. I mean, a DAC column, they're like, no way. But it's a necessary evil. I don't know what word I want to use for it, but it's a very popular technique that's growing for maybe the first time since insulin. Because of its growth and because it's just been sort of a compensating behavior in industry for some time, I think it's going to grow, and you're going to see major advancements in that in the next few years.
Better, cheaper, or faster, but not all three.
You can't get all three, right?
Business, right?
That's the rule of you pick two is what I always said. Pick two and go after that. That's an area, again, we're focused. I wish we had more resources. I could build an entire team around that if people let me. But at the same time, we do have a team and a lot of knowledge and customer collaborations that we're working with in order to provide those as well.
Very good. Sean, I really appreciate your time today. I think this was a great introduction to the chromatography audience in terms of what Phenomenex is working on, and I hope to have you again.
Hey, Dave.
Again sometime.
Thank you for having me. Pleasure chatting with you. And I look forward to chatting about this and other topics in the future.